one last thing on proteins, and that is what happens if the proteins get misspelled. So the information for proteins comes from the last group we're going to talk about here in a second, which are the nucleic acids. And sometimes the directions are written down wrong, and you end up with an incorrect amino acid sequence. So if you look down here on the picture, we've got correct spelling, protein, and then the misspelled version, which spells plotein, right? So the R got replaced. What happens is if a protein gets misspelled, it changes that 3D structure most of the time, and it changes the active site. So the active site, or that open Pac-Man part where the substrate comes in and fits, will change. And so in this case, with this enzyme in this picture is taking phenylalanine and breaking it down into tyrosine. The active site is required right here in the center. You can see it's very tight and small. But if it's misspelled, then the active site is open. And so in this case, phenylalanine can't attach to the enzyme and won't get broken down. In this case, with this enzyme, phenylalanine will build up, and in this condition, phenylalanine building up is actually deadly. This is a genetic condition that can lead to death. Now, in this case with phenylalanine, you can actually control the amount of phenylalanine that you are consuming, and it won't affect you as long as you control that amount, because your body can flush out the extra phenylalanine. But there are other non-functioning enzymes with these active site disruptions that can cause problems. Some of them can lead to death. Some of them can just be uncomfortable. So for example, why do some of us get sick when we drink milk or eat dairy products? Well, it has to do with the active site on lactase. So lactase, you saw it, I remember right in the video, just a little bit since I've seen it. So that active site doesn't fit, and the person cannot break down that lactose sugar, and that can actually lead to indigestion. Depending on how well this works, it may be able to break down some of it. You may have one functioning copy of the directions, and so you might be able to make some functioning copies of this, but as long as the active site is not the correct shape, you'll end up being ill. So I've got this question. Lactase cannot function. Lactase, again, is the enzyme. Enzymes end in ase. It can't function properly in this example because of A, the shape is incorrect, B, the amino acid sequence is incorrect, C, the color is incorrect, D, both A and B. And in this case, it is D. Both A and B are the correct answers because the active site is incorrectly shaped, and it's incorrectly shaped because the amino acid sequence is incorrect. That brings us then to our last group of macromolecules, and it's the nucleic acids. So the nucleic acids, their function is to store information on how to build and run organisms. So all living organisms have DNA. Even the dead ones do, but all organisms are going to have DNA, and they're going to have RNA. Those are the nucleic acids. So these are the macromolecules that store information, and they're going to store the information in the order of their nucleotides. So just like amino acids make up proteins and monosaccharides make up carbohydrates, nucleotides are the building blocks, the pieces for making nucleic acids. So what we're looking at here, we've got strings of nucleotides. An individual nucleotide is right here. It's a phosphate, a sugar, and then a nitrogen base. The nitrogen bases can change. So you can have this guy, you don't need to know them by their full names. I'm just going to be asking you to know them by their initials. So we've got G, C, A, and T. All right. And so adenine, thymine, guanine, cytosine. So A, T, C, and G for those for the nucleotides for the nucleic acids. There are two types of nucleic acids. I just mentioned those a little bit ago. So there's DNA, stands for deoxyribonucleic acid. DNA is fine for this class. And then RNA, which is ribonucleic acid. We'll see in a little bit why they have those different names. And both of these are essential for making proteins, right? So they store genetic information, and then, like I said, they're in charge of storing the information to run and build a body. And we just saw in our last lectures that proteins make up most of an organism. And so these guys are going to have the directions and be in charge of making those proteins a reality. So how do they store their information? Well, they store the information in the sequence of those bases. So the A, G, C, and T's, the order of those letters is what gives us the information. So if one of these letters is incorrect, gets swapped out, gets taken out, or a section of them gets taken out, it changes the direction and can change the type of protein or the shape of the protein that's being made. So DNA, we're going to talk about it first. It is the holder of the genetic information. So it's going to hold all of the directions on how to build and run the organism. It's going to use those four letters, A, T, C, and G. It's going to hold it in a structure that we call the double helix. Why do you think it's called a double helix? Take a look at the picture. It's a double helix because there are two strands. So two strands of nucleotides that are stuck together in the middle. And the helix part is the spiral part. So helix just means spiral. The pairs always pair together in the way that you see them. A to T, C to G. And I always remembered that they pair this way because the C and the G look alike, but I actually just heard a new one the other day that I think is kind of cool. Apples are found in trees and cars go in garages. So whichever one helps you remember which one goes with which. But you can see here they pair up across from each other to make that double strand. So when you've got a strand, you can, if you've got one side of the DNA strand, you can find what we call the complementary strand, basically the matching strand on the other side. So take a swing at what the complementary strand would be for this. So what would match this on the other side if we had, let's say this is where that DNA was attached. What would the letters be that would sit right on this side? All right, and I'll start it off. It should be C, C, T, A, and so on. And according to the pictures we just saw, they would be bonded across here. Okay, so got another one here. So what's the complementary strand to this strand? And the clue to look for is you've got CCCC. So you're going to look for one that starts with GGGG. So you can leave out A, you can leave out D. You know it's got to be one of these two. So then the next thing you just need to look for is, do the next two letters match the way they're supposed to? It should be AA. This one's TT. Can't be that. So it has to be C. All right, so how is DNA different from RNA? I'm sorry, how is RNA different from DNA? So RNA is a translator. It's going to, in a couple different steps that we'll talk about later in the semester, read the information from the DNA. So you can see that over on this side. So it's going to take the information from the DNA, we get RNA, and it's going to use that information to direct the protein production. So we'll talk about that in more detail in another chapter, but that's RNA's function. It's the middleman. It's taking information from one place and taking it to another place. RNA is a nucleic acid, so it's built out of nucleotides, but there are some differences, and I've got that over on this slide right here. So one is the sugar molecule of the sugar phosphate backbone. So if we go back over here, the sugar phosphate backbone is just where the nucleotides attach. So it's got a different type of sugar. This is actually why it's called ribonucleic acid, because this is the sugar ribose. And DNA is deoxyribonucleic acid because it's deoxyribose. So different sugar. The other thing is that RNA is a single strand, so it's just one string of nucleotides instead of the two that we see with DNA. And then we're going to use uracil, U, instead of T. So there's no T in RNA. So if you're looking at a string of letters and it has a U in it, you know you're looking at an RNA sequence. If it has T's in it, you know you're looking at a DNA sequence. So the last thing I've got here for the macromolecules is taking a look at an RNA sequence that was made from a DNA sequence. So here it pairs the same way. So we've got actually the same DNA sequence from the last multiple choice question, but now you're going to pair it as its RNA. So the pairing rules are the same. You're still going to pair C to G, and you're going to pair A to U. All right. So you're going to look for any answer first that has a U in it. Okay. So if you look first for ones that have U's in them, then you can tell right away that A and B are out. Those have T's. Those have to be DNA molecules. Then you just need to look for the one that matches the one that you're looking for. So you know that your answer has to start with 4 G's. C doesn't, so it's out. It has to be D, but we'll double check to make sure it's right. So D, or G, 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 yep, because it's got C, 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 C. Then we've got 2 T's, so T still matches with A, so A. Then we've got an A. A would match with T if it was DNA, but now we've got U, right? Because this would be a T, but it's actually a U. Then we've got A. Oh, it looks like I have a typo. So technically, if you answered nothing, you're right. So let's write this one out. So I can't believe I did that. G, A. Now this would be a T if it was DNA, but it's going to be U. Then it would be C. Then we've got A again. It would be normally T, but it's going to be U, and then G. All right. And if you would like some extra practice on going from DNA to RNA, DNA to DNA, just let me know and I can point you to some different sources.